This tutorial demonstrates how to navigate and mine the data available from the visual coding dataset in the Allen Brain Observatory. This dataset is a physiological survey of neural activity in the cortex of an awake behaving mouse during visual stimulation. Mice genetically encoding a fluorescent calcium indicator, GCAMP6, were stimulated with 5 synthetic and natural stimuli. Cells were imaged using 2-photon microscopy in 6 distinct visual areas of the cortex. The different transgenic mouse lines were chosen to visualize cells in specific layers of the cortex. The cells were imaged at different depths to capture the subsets of cells labeled in these transgenic lines. This tutorial will cover how to search and download the data available from this dataset. Clicking on the Experiments tab will allow you to search and mine the cell population data available from each experiment. You can mine the individual cell responses from the entire dataset by clicking on the Cells tab. The Allen Software Development Kit, or SDK, is a Python-based resource that serves up source code to read and process the Allen Brain Atlas data. Programmatic access to the data is available by clicking on the SDK tab. More information about the characterization of the transgenic lines available in this dataset is available from this tab. The Documentation tab will take you to our white papers describing the methods, as well as supplemental data important for characterization of the transgenic mice. The online help is your first stop if you get stuck navigating the site after this tutorial. Your first step in understanding the data representations is to view each of the visual stimuli pages, where we describe the visual stimulus, including a demonstration of what it looks like and how it was presented to the mice. This page also describes how the data were analyzed and the rationale behind the visualizations we created to illustrate the cellular responses to each stimulus. Each stimulus page also has an interactive example of the representation. Hovering your mouse over the different aspects of the visualization will bring up the stimulus described by that cellular response. Analysis of this data can either be carried out at the experiment level, if you're interested in population responses, or at the cellular level. We'll first dive into searching via experiment. Clicking the Experiments tab will load all the data with one experiment per row. You can search by subregion of the visual cortex or by brain area, imaging depth, or by Cree driver line. To see where GCAMP6 expression in the different transgenic lines is enhanced, click on the Transgenic Characterization link for a description of where activity is likely to be imaged. Each experiment has a unique experiment ID and is described by the experiment parameters, as well as several population statistical measures that are summarized by a suite of thumbnails. The columns to be displayed can be managed from the drop-down menu in the top right-hand corner of the Filters box. To see the cell responses in an experiment, click the View Cells link. To dive deeper into an experiment, click anywhere in the experiment row to be taken to the Experimental Detail page. This page summarizes the population responses of the cells in a given experiment. At the top of the page is metadata for the mouse, the area, and the depth of the experiment. Each experiment is composed of a series of 3 one-hour-long 2-photon calcium imaging sessions on an awake mouse exposed to a series of visual stimuli. Each of the 3 sessions is laid out as in this figure. Each stimulus is color-coded, and note, not every stimulus is presented in each session. A composite view of the cells from each of the individual sessions shows the cells imaged in this cortical view. Also included on this page are thumbnails that summarize the responses of the population of cells in this experiment. Beside each thumbnail is a description of how the feature was calculated. If you're less interested in mining the population responses of an individual experiment and more interested in mining the cell responses from all the cells in the dataset, you will want to use the cell search features. You can search for all cell responses in the database by clicking on the various interaction points on the landing page, either the transgenic mouse lines, the visual areas, the depth indicators, or the visual stimuli responses. If you're interested in specific cell response features, click the Cells tab. From this page, you can search for cells that show specific responses to the various stimuli. By default, the cells are sorted based on the significance of their measured responses to presentation of the static grading, listed here as their p-value, but can also be sorted by several other parameters from the drop-down menu. The sort filters displayed are dependent upon the visual stimulus. Once you have selected a group of cells to explore further, there are a couple of ways in which you can download these cells for analysis. Clicking the Download All Data link will download a CSV file with the computed features and metrics of every cell in the database. This file also includes all the information the interface needs to produce the web page, so the columns related to visualization image paths can be ignored. Clicking the Use the Current Filter with the SDK link will take you to a page with a snippet of code which will enable you to download just the data for the cells you're interested in. Briefly, you will need to create a Jupyter Python notebook and install the Allen SDK. Instructions for this are available from the SDK tab in the banner menu. From the Install Guide, you can install the Allen SDK. And once you have installed the SDK, run the code from your search. You can visualize your matrix using the Pandas library, and you can export to a CSV file. If you'd like to compute your own metrics, work with the fluorescent traces, or analyze the eye tracking data, the ideal way to interact with these data is directly from the Allen SDK. To get started, read through the information on the Brain Observatory from the SDK page and download the Jupyter Notebook, which will provide the code for downloading and analyzing the data available from the Brain Observatory. Should you have more questions or would like to provide feedback, please send us a message from the link at the bottom of any of our pages and we will get back to you. Thank you for taking the time to view this tutorial.